Okay. So unlike carcinogens, where pretty much no one will disagree that a carcinogen is a chemical or agent that can cause cancer, endocrine disrupting chemicals don't have a unifying definition across world organizations. And so I like to use the definition by the Endocrine Society, which is a professional organization of clinicians and academic research scientists. And they define an EDC as an exogenous or non-natural chemical mixture of chemicals that interferes with any aspect of hormone action. And then there was a slash on my title today for MDC. So this is a key characteristic that an expert group is convening to discuss and develop in September. And so I'm gonna try to show you some of my emerging thoughts about that in the context of this talk as well. And metabolic disruptors are similarly not defined in a kind of universal way, but there was a Parma consensus statement conducted by a group in Parma, Italy, including myself, stating that it's an exogenous chemical or chemicals that play a role in altered susceptibility to obesity, diabetes, and related metabolic disorders, including metabolic syndrome. And so really, in a way, it's going to have some overlap with hepatotoxicity because some of these metabolic disrupting symptoms have to do with fatty liver. Obesity is often considered a chronic low-grade inflammatory state, so there'll be aspects that relate to inflammation and the immunotoxicant story. And then a lot of these are risk factors for cardiovascular disease and have overlap there. And so I'm going to talk to you a little bit about the cardiovascular cases today too, just to touch on them since you won't hear about them today otherwise. So I like to think of this marathon of KC development that's been occurring more recently in light of California's hazards trait legislation, where the hazard traits are basically all the health endpoints that are relevant to the people of our state and the design and implementation of programs of toxic chemicals. And so within the concept of toxicological traits, we've got hazard traits relating to the reproductive system, endocrine systems you've already heard about, and then the cardiovascular. So a few years ago, gosh, 4 now, we had a meeting, actually, was it right here? Yes, of experts to explore the idea of advancing the key characteristics framework to include reproductive toxicants and endocrine disrupting chemicals. And we ended up breaking that off into both male and female reproductive cases separately. And I'm, of course, going to tell you about the key characteristics of endocrine disrupting chemicals. And so just a snapshot of what's out there already. So neurotoxicants or developmental neurotoxicants is the only one that I think hasn't been touched on during this meeting so far. That's also being written up to be submitted. So when we thought about the endocrine disrupting chemicals and how they might be influencing or what their key characteristics are rather, because we don't have this unified list of 100 monographs to reflect back on, like, we really started it as a thought experiment of what do we know about the way the endocrine system works, and with the notion that chemicals can interfere with each step of the process. And so we can think about it really from like kind of a life course of hormone action, if you will, where, this is our circle, and a hormone starts out being synthesized, and that synthesis can be modulated by an EDC. The hormone is transported. Sometimes it's immediately transported as soon as it's synthesized. Sometimes it's stored for later, like as thyroid and insulin are. Then it goes through our blood and distribution, or in circulation, depending on what your training background is, how you would phrase that. And ultimately, would get broken down. So we would no longer need the hormone at a given time, and it would somehow be deactivated or in this kind of way. And I'll skip over 10 for a moment. Then when the hormone is in distribution, it can also encounter a receptor that it binds to, and it can activate that receptor. Usually doesn't antagonize it, but an EDC can do so. The receptor expression itself, so being able to encounter that receptor either in overabundance or underabundance, can occur. That receptor usually has some kind of signal it transmits, and so that signaling can be impacted. A number of the endocrine hormones are binding receptors that are nuclear receptors, and so part of the machinery that creates their ability to signal includes epigenetic alterations, basically, classic transcription factor regulation aspects of things. And then, 10 out here is really about this concept that Dory mentioned, with this idea of like developmental susceptibility and this interest in capturing the fact that we know that certain life stages are more at risk of the effects of exposures. And so looking at cell fate in terms of what does the cell end up becoming when it's a differentiating pluripotent cell, and maybe that might include modulating proliferation or its death, including a type of death like apoptosis. And so I've got here the little diagram that I kindly received from Kate when she was at IARC, because kind of the IARC framework, which is how I like to think of utilizing the KCs, where in our paper, we have a couple of examples of endocrine disrupting chemicals, but I've mapped some that aren't in the paper so that you can have something that makes this talk worth listening to apart from just reading my paper. And what I've done is I've mapped, or I've pointed out where I think we might have some overlap with metabolic disruption and BOLD, to kind of highlight how that might be emerging out of in the future. And so to give an example here, for endocrine disruption that overlaps with metabolic disruption, if we look at the phenotype in humans, we know that humans have increased risk of obesity, particularly with DDT chemicals that are found in utero exposures, and this has been shown in numerous meta-analyses. And so that data is quite strong, and it's also been shown in several rodent species. And so we can think about how this mechanistic data would be integrated with that potentially. DDT and its metabolite DDE have been known for some 50 years to bind estrogen receptors and activate them, and they can also antagonize the androgen or testosterone receptors. So there's also this like kind of classic endocrine disruptor concept that we all think of generally when we hear those words. So I'm telling you about kind of more of a novel aspect of my own other research area. But related to obesity, we also know that DDT can influence hormone receptor expression. For example, the membrane expression of TSHR, which is part of the thyroid hormone axis, and thyroid hormone is very important for energy balance. And DDT can alter the signal transduction in hormone responsive cells, not just with estrogen, but with signaling, which has been very, very well documented, but also related to insulin signaling. And this applies to mouse liver studies. So we've got some hepatotox there and a few other examples here as well. These highlighted examples of epigenetic modifications are actually some of my unpublished work, so I'm not going to get into them since I know we're behind schedule. But the next half of the KCs, you can see that now that I've shown you all 10, that DDT and its metabolites map to every single one of the endocrine disrupting key characteristics. So I hope you can appreciate that it is kind of a classic EDC. So yeah, DDT and DDE can alter hormone synthesis. This deiodinase is really important for the synthesis of thyroid hormone, and that's been shown in 2 rodent species. Also hormone transport, where insulin secretion by the pancreas has been shown. And the distribution of hormones, we really like this one because it's something you can measure in humans a bit more easily as kind of a biomarker. And we have seen increased circulating insulin levels consistent with risk of type 2 diabetes in the overall context of that study, and problems with decreased expression of thyroid hormones, and thyroid hormones are involved in causing us to burn calories so that we don't store them as fat. And then, of course, some influence on estrogen metabolism and can change the fate of cells. So in the context of this like metabolic disruption, DDE has actually reduced the area of a pancreatic beta cell, which is where the insulin is secreted. So sometimes that happens when you produce too much insulin, it causes your beta cells to die. And when you have all your beta cells die, that's when you develop full-blown type 2 diabetes. So several aspects of that pathology have been documented. I can also do the same thing with the cardiovascular disease toxicants. And just taking a look at the clock. I do want to take a moment here to point out that, yes, I just showed you an example where all 10 KCs were occupied. And we heard other examples where there was many KCs occupied by a chemical, but it's not just to count them and decide. There's no magic threshold. So in our manuscript, we also show that the key characteristics for perchlorate, which is known to cause hypothyroidism, is just one single key characteristic, and that is to disrupt the synthesis or production of thyroid hormone. And so I think that that's important to remember across all the key characteristics, that it's not just the count of key characteristics, but also the quality of the data. And in the context of that perchlorate example, there's really compelling data in humans that helps justify that singular key characteristic being of sufficient nature. So I'm bringing up cardiovascular disease because I have shown in my mice that we have increased cardiac hypertrophy in my mice that are exposed to DDT at levels relevant to the human condition. And a collaborator who's actually the lead author on the key characteristics of cardiovascular cases, Dr. Lars Lind, we also showed that in his human study of 1,000 elderly people. And we went on to show that that was actually primarily mediated by the relationship of DDT and obesity. So meaning that DDT as an obesogen can then in turn increase risk for heart cardiac hypertrophy. And so in this example, you can see it's much less studied. They're not meta-analyses, they're just two anecdotal, one human, one rodent study. And when we map the mechanisms, this is some interesting mechanism I probably don't have time to explain to you, but it's not related directly to the phenotype, but it is related to a cardiovascular key characteristic, of course. And then again, the metabolic disruptor kind of overlapping ones are in bold here to show you kind of how that, that interplay potentially works. And you'll remember that we saw impairs mitochondrial function. So we saw oxidative respiration impaired in mitochondria across tissues of cows, rats, and mice. These tissues included the heart and also the liver. So one could map this directly to the hepatotoxicants that you heard about earlier today. And then the autonomic nervous system activity. This one is one of my favorite parts of my research, where we're showing that the DDT is reducing how we burn calories because our nervous system isn't able to send those signals into the fat burning machinery and calorie burning machinery of our bodies. And then you can see that alters hormone signaling is actually built into the cardiovascular disease key characteristics, as is inflammation. And here, unfortunately, oxidative stress is 10, not 5, but we get the point. It's also contextual. But this was considered strong evidence in the IARC monograph that reviewed DDT as a 2A carcinogen. And so I just want to show you this box as we transition into the next session after a brief break. What we suggested were recommended uses of the key characteristics of EDCs. I'm not going to read through this for the aspect of time, but I do want to point out that some of these points were really made quite well by Kate earlier. This part I just want to explain because I think it's kind of awkwardly written. The resulting included papers can be further evaluated based on design and reporting features as defined by the end user. So for example, if you're an agency that doesn't define an endocrine disrupting chemical as having all 10 of those properties, and you wanted to use the KCs, you could simply choose to use a subset of the KCs for your personal work. And so however you choose to scope it, that would help you decide how to use the KCs. And I think that's an ongoing theme among people who are trying to implement the KCs for their use, is that how you use the KCs depends on what you're trying to do with them and to achieve. So you could use the KCs to organize your information and then plop them into an AOP, an adverse outcome pathway. I would never do that, but if you needed to do that because of a regulatory necessity, that could potentially be a way that they would interact. And we favor the idea of working towards having a concept of strong, limited, or inadequate to describe the weight of the evidence from these cases, similar to how IARC does with the carcinogens. And I think the fact, and this was a point that Dave was making earlier, that because we're not trying to draw arrows specifically from the toxic substance to the adverse health outcome, that it allows us to just fill in parts of the table and empirically over time see how that all fits together with what we know. And I think that really is very important and practical, especially when you're dealing with data sparse chemicals. So with that, I just want to thank the co-authors on our endocrine disruptors consensus statement. You can see many are from the US, but we also have Europeans, involvement from the OECD, Cal EPA, US EPA, IARC. And then this is the entirety of the EDC group, as well as both the male and female reproductive toxicant groups from 2018. So thanks so much for your attention. When do you want everyone back? Okay, 10 minutes, so that would be at, let's call it 4:07. 4:07, I'm watching you. Okay, thanks.